Unknow.
Hello, I'm Manuela Casas. I'm a PhD student at the Laboratory of Sensory Transduction and Nociception. And, well, I'm an early career scientist, and I'm still super humbled by the talk earlier given by Nelia. But still, I wanted to structure this talk as also a journey into time. So, beginning with a little dive into the past, I was born and raised in Madrid, and I've always been into science. So when I decided what to study, I went for biology at the Universidad Complutense de Madrid. And within that degree, I had the opportunity to go on an Erasmus stay at the University of Helsinki. And it was there when I really discovered neuroscience through some courses that I was able to do. And I was super excited about it, so when coming back to Spain and finishing my degree, I went for a master's degree in neuroscience, which I did at the Universidad Autónoma de Madrid. And in order to complete that master, I did my master's thesis at the Instituto Cajal in José Luis Terjo's lab that some of you, I don't know, might be familiar with. And I think the mic is off, but I don't think you can hear me. I absolutely loved that experience. That was my first real lab experience, if you will. And when I finished that, I was convinced to give that science path a try. And that was when I discovered Felix Liana's lab here at the Institute. And I got accepted with an FBI grant, which takes us to the present, to now, what I'm doing here at the lab at the Institute. Well, the big heading that you're seeing right there, the molecular diversity of thermoreceptors, mechanisms of cold-induced pain in a neuropathic pain model, is a super long name. It's the proposed title for my thesis. And let's try to understand a little better what is this about, what is this project about, and where am I at the moment. So, for giving context, the laboratory I'm working in, as I said, is the Lab of Sensory Transduction and Nociception. The name is quite self-explanatory. We work in the sensory processing of the dorsal root ganglia and also in the spinal cord. And the lab has an expertise in functional characterization of sensory neurons and their receptors, not only in physiological conditions, but also in pathological conditions. So within that background, what am I doing to study this molecular diversity in a neuropathic pain model? Well, the first thing that I need is a neuropathic pain model, obviously. So among all the models that there are, what I'm working with is the so-called chronic constriction injury. I'm working in mice. And this model consists in cuffing the sciatic nerve with a plastic tube. This makes a constriction that is compressing continuously the sciatic nerve. And this model has been validated to mimic pathologies such as the sciatica or the carpal tunnel syndrome that some of you might be familiar with. I hope not, but it's quite common. And within those pathologies has already been published that there is an increased sensitivity to cold, so it makes sense to study the affected neurons that have a role in the cold sensing project. Also, the model mimics well this increased sensitivity to cold. So, for studying the cold sensing project, we need a cold receptor in those neurons. And among the receptors that are reported, I'm focused on TRPM8. TRPM8 is an ion channel that not only detects cold, but is also activated by several other agonists, such as menthol or immunosuppressants, among other molecules. This channel has been reported to have a role as an analgesic with inflammatory states, and this is one of the reasons why we can apply cold when we have an inflammatory injury, but has also been reported to be negatively modulated by inflammatory mediators. So it clearly has a role when has some changes when some injuries are happening. Okay, so I have my receptor, I have my pain model, but how do I detect the neurons that have this channel in order to study them? Well, I'm using a transgenic line that has an insert of YFP within the TRPM8 gene. So the neurons that express the gene, if given the right light stimuli, will shine with fluorescence. So positive neurons for TRPM8 will shine, so I will be able to detect them. And with this line, it has already been reported that the cells that are positive for this channel can be divided into two different populations that are different one from the other in size, having one that is smaller and shinier, and also functionally, because these smaller and shinier neurons also respond more when given a stimulus versus the other population which is made of bigger cells that are also less shiny and that with that very same stimulus respond less. So this indicates us that probably the amount of the channel that these two populations have are different. Okay, so I want to study within these two populations of neurons what happens when the animal undergoes this chronic pain condition. So in order to structure that work, the workflow that I go to is basically briefly the following. First, I take my mice and I apply some behavioral tests before and after the surgery to see if the surgery is being implemented and is actually making the mice develop that chronic injury state. And 15 days after I do the surgery to the animals, I extract the ganglia that innervate the sciatic nerve, so I'm able to isolate the neurons and go for the molecular characterization. Okay, so regarding behavior, where am I so far? I'm doing two different tests right now. I test the mechanical sensitivity with the bone freight test. Maybe some of you are familiar with, but for those who may not. This test consists in applying a mechanical force to the paw of the animal and measuring how much the animal reacts to that stimulus. So the more the animal reacts, the more sensitivity. If the animal is not moving the paw, it means that it's not even feeling the force. And to detect the cold, for the cold sensitivity, I'm doing the dry ice test, which is a very similar approach, but instead of a mechanical force, I put the mice on a glass surface and I stimulate them with a cold source to the paw. What I measure here is the time that the animal needs to retract the paw. So the shorter the time, the more sensible they are to the cold. And by now, the results that I have so far are looking quite solid. In the red lines you have the constricted mice, and the black line will be the sham controls. So after I do the surgery, they develop both mechanical sensitivity and cold sensitivity compared to the controls. So things are looking good for going to the molecular assays. So before diving into that, I wanted to give a brief explanation of how I obtain my samples for the molecular assays, because I'm sure that many of you are familiar with FACS sorting, which is like the go-to protocol, I would say. But I'm not doing that right now. I'm doing another alternative method, which is called cell picking, and it's also self-explanatory name. I pick the cells. So what I do, as I said, I extract the ganglia, and I do a cell culture, and I plate the cells onto a cover slip, which is a tiny glass surface that I can put under the microscope. So within the first culture, I can put with a fluorescent microscope, I can detect my fluorescent cells that, if you remember, are the TRPMA positive cells, and detect them, and with a glass pipette, a pipette that is quite similar to the pipettes used for patch clamp, but slightly bigger, I approach the cell and I manually extract them. I suck the cell out of the culture. So I put each cell into a different tube and I immediately freeze them so they don't degrade, and I can go for the molecular assays. So for the molecular part, which is like a big part of my project, I'm doing right now two different approaches. One being single cell. With this method, I have a lower amount of cells as if I was using FACS sorting because of the manual restriction that I have with that technique. But that allows me to go for a kit that allows maybe a little bit more depth of sequencing. And right now, I'm in the analysis part of the results. I've already run the experiment, but the results are not ready to be shown. And in parallel to that, I'm also doing nested qPCRs with the same obtaining method. So I'm also working with single cells, and this allows me to validate the results of the RNA-seq for a smaller and also cheaper method of testing those set of genes of interest that I'm working with that are appearing in the RNA-seq. But I'm not showing results of this part because it's a work in progress. I'm in the middle of that, and as I said, like, results are not ready to be shown. I hope to come back soon-ish, at least, to show them so we can all discuss them. But that's already looking into the future. So what to expect in what's coming? Because yeah, I'm just starting. So if we divide future into short term and long term, and short term being everything that becomes before finishing the PhD, although that might be still a few years. I have a bunch of goal sets. First of all, completing the molecular characterization, and that comes with a bunch of experiments. So trying to not get overwhelmed with that. But not only the molecular aspect, I also want to implement a functional characterization of that model. I wanted to stay abroad before finishing, and I'm also sure that before I finish, more experiments that I don't even have in mind yet will appear, as it's quite common in science. But that still is not a full dive into the unknown as compared to what comes after the PhD, which is quite scary sometimes, and I think that many of my PhD student peers can relate to this, maybe not fear, but stressful thinking into what comes in maybe 5 or 10 years ahead. We don't know where we'll be, maybe academia, maybe not, maybe science at all, maybe not. We, not only PhD students, but I guess like everybody in the audience already knows how hard science can be, how stressful deadlines are to meet sometimes, and how it's quite easy to forget why we came here in the first place. And that's why I wanted to take this final look into the future with some of the eyes of the past, if you will. So maybe you have noticed my clothes today. Pretty cool, right? And you might think that I'm looking a bit childish with this dinosaur overall. And yeah, I might be. But in a day like today, I wanted to just remember that little girl that was excited with science, that was trying to do these little chemistry experiments at home, in the kitchen, that was loving the science courses at school, that chose biology, and ultimately came here. Because when I was a girl, I didn't know I would end up here, and I would say I would be super stressed if I knew that I would end up here. So for a moment, let's forget about how stressful life can be and try to bring back all those little girls that are excited and celebrate that we are here and how far we can still get. And just to finish up, I wanted to thank my lab and all its members, and also all the facilities at the Institute that make this work possible, as well as the funding that, of course, make all this work possible. And you for listening. Thank you.
Thank you very much, Manuela. Questions, comments?
Thank you very much, Manuela, for your presentation and for being brave today to share not only your data but also your feelings about how you are moving forward into the future. I have questions for the science part and then a comment. So I think because today, is the day of women's science, I feel like asking if you have noticed any difference, sex difference in your behavior with the pain threshold being different for males and females, and if you take this into account in the data.
I study with males and female mice, but so far the responses are pretty much the same. I believe from what I'm seeing in my results that the injury itself is strong enough to make all those possible sex differences I don't know, like be secondary to that. So what I observe is that when I do that constriction, regardless if they're males or females, I obtain the same response. But yeah, with the RNA-seq, I also have like my cells labeled as which cells come from males and which come from females to check if some possible difference may show up.
Yeah, so that's another question that I was not very clear on. You know, we're not used to this protocol or like the cell picking. So I guess that you have the cells extracted in the petri dish from male or female, and that's how you can tell, right?
I mean, I take like each animal with their own like setup. So I take like I know one animal one by one. So I have like a female mouse, like everything's labeled. So like all the cells that are in the culture are for that mouse. And like even if it's the same group, so even if I have like 3 females with the injury, I go one by one because of the setup needs. Yeah.
I mean, in the cell picking technique, from what you just showed, it seems that perhaps the contrast of the fluorescence, when you're trying to pick, I imagine the cells that they are expressing the receptor, it's not.
No, the picture is quite bad, I know.
I don't know, but I was wondering about the weak fluorescent cells. For you to be able to collect that sample, it is, I guess, I don't know, it seems to be perhaps a little bit more difficult than far sorting.
The intensely fluorescent cells, the picture is, it's easier to see through the microscope than with the picture that I showed. It's not the best picture. It's I did it with my phone, so it's not it's not a pro picture. But I still wanted to show you how the pipette looks and approaching the cell. So they are showing better under the microscope. The intensely fluorescent are super easy to recognize. The weaklies, yes, sometimes. I mean, if I'm not sure if it's shining or not, I don't pick that cell because. I mean, I don't pick so many, like it's an experiment, a whole day experiment, and then the culture starts degrading because it's under the microscope light. So I have limited time to do the experiment and also like limited time, like me being able to do the experiment. So I don't pick like every cell that there is. So I go for like the most clear ones. Still, yeah, there's we're having some debate also by seeing the results that maybe some of the weakly fluorescent cells might or maybe not from that population, because yeah, if they're shining maybe a little bit, maybe they don't have the channel, but it's an artifact from the preparation, and yeah, that might be. Yeah, that's something that's been on my mind as well. So.
Okay, well, thank you.
Thank you. One question? I cannot resist. So first of all, I didn't think that your outlook was childish. I just thought it was cool, so I will wear this every day.
Thank you.
Maybe it's not representative of no, I have a comment because we have been talking with you. We have been talking about the fact that, for example, the fact that sometimes PhD students don't feel motivated enough or satisfied. So I think it's very important to think about how excited you were as a child or when you were younger. But I've been reflecting on this, and maybe it can also be that you're lacking role models of people that look happy in their job. Yeah. So I've been doing autocritic, and maybe I also with my students share all my, I don't know what I'm worried about, not getting a grant, not getting a position, and I fail to share how cool it is what we have done. So basically my question is, do you see us, emergent researcher or senior researcher, do you see us happy?
I mean, I think it's like from my personal point of view, I think it's important to show like the ugly part because it's a reality. Like we all get stressed, we all need help sometimes. So I don't think we need to hide that. But I think you made a super strong point on like also maybe we need to encourage also that positive things that as these talks today to celebrate what we do, to to just think of what we've achieved and what's to come yet, because that's also a source of motivation. And maybe if this talk leads somebody to think about their own path and, oh, how cool would it be to do blah, 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 or whatever. And I think that maybe you, like in the more senior positions, if you encourage us to do that as well, that's super helpful, because it's also nice to see, like, to celebrate the achievements and also to show that not everything is a deadline and not everything is an evaluation and not everything is an exam or whatever, but also like why the science that we do, that we like it.
Thank you very much.
And also you left like comment on generational comment or difference, maybe what you were mentioning Sylvia, or I don't know if it's a difference between generation or difference between two stages in life. But since we are, you are totally entitled to do this because you are also the representative of this student, one of the representatives in the institute. But since today is also the day for women and girls in science, I don't know if in this discussion that you have with your peer, you think that there is, the comment is different between girls and boys? You still, how present is the sex factor in this conversation?
But I'm not super sure if I got like the question.
If you believe that these worries are different between
okay.
boys and
I wouldn't, again, I can just like speak from my personal point of view. So like talking with my peers, I guess like probably some others have other opinion. But I haven't met like this strong bias between boys and girls. I think that regardless the gender, if we all think about like mixing personal life with science, maybe starting a family or things like that, are quite a challenge. But so far at the stage in my career that I'm at, that most of my peers are not having kids yet or anything like that, I'm not finding that bias. I don't know if maybe a few years ahead, if, I don't know, but if me or any of my peers, female peers, wanted to have kids, maybe that would lead to a bias in how we see the science life. But right now, I wouldn't say that it's a strong observation.
Thank you very much, Manuela. If there are no other questions, we move to the